Semen and sperm, are they actually the same thing? Not even close. One is a specialized reproductive cell carrying genetic information, while the other is a complex biological fluid designed to transport and support those cells. In this video, we're going deep into the science behind both. Welcome to Zenvy Health, where we break down human biology and medical science in a simple but clinically relevant way. Today, we're exploring one of the most commonly misunderstood topics, semen versus sperm. Understanding this distinction is essential for anyone studying human reproduction because sperm and semen have completely different origins, structures, compositions, and functions. Let's start with sperm. A spermatozoon is a highly specialized haploid male gamete produced in the testes through a process called spermatogenesis. Spermatogenesis takes place inside the seminiferous tubules and involves a sequence of cellular events, beginning with spermatogonia and progressing through meiosis to form haploid spermatids, followed by spermiogenesis, during which these cells develop into structurally specialized spermatozoa. After leaving the seminiferous tubules, sperm pass into the epididymis, where they undergo important maturation and acquire progressive motility and functional competence. A mature sperm cell has three major regions: the head, middle piece, and tail. The head contains the nucleus, which carries the paternal haploid genetic material. Its anterior portion is covered by the acrosome, a specialized structure containing enzymes involved in interactions with the oocyte during fertilization. The middle piece contains numerous mitochondria arranged around the axonemal structure, providing ATP required for sperm movement. The tail, or flagellum, produces the motile force that allows sperm to move through the reproductive tract. Now comes the important question: what is semen? Semen is not simply sperm. It is a complex biological fluid containing spermatozoa mixed with secretions from the male reproductive tract and accessory sex glands.
These secretions provide nutrients, enzymes, electrolytes, proteins, and other substances that help maintain sperm function and facilitate their transport.
A major contribution comes from the seminal vesicles. Their secretion forms a substantial part of seminal fluid and contains fructose, which provides an energy substrate for sperm metabolism. The seminal vesicles also contribute other components involved in the physiological environment of semen. The prostate gland adds another important component. Prostatic fluid contains enzymes and other substances that contribute to the biochemical properties of semen. One clinically important enzyme is prostate-specific antigen, or PSA, which participates in liquefaction of the seminal coagulum after ejaculation. The bulbourethral glands, or Cowper's glands, contribute a smaller amount of mucus-rich secretion. This secretion helps provide lubrication and contributes to preparation of the urethral environment for the passage of semen. During ejaculation, sperm stored in the male reproductive tract are transported through the vas deferens toward the ejaculatory ducts. As sperm mix with secretions from the accessory glands, the final seminal fluid is formed and is expelled through the urethra. Sperm and semen perform different roles. Sperm is the actual reproductive cell whose primary function is to deliver paternal genetic material to the oocyte. Semen is the surrounding fluid that transports sperm and provides a supportive biochemical environment. This distinction becomes especially important clinically. A person can produce semen but still have abnormalities in sperm concentration, motility, or morphology. In semen analysis, several parameters can be evaluated, including semen volume, sperm concentration, total sperm number, motility, morphology, and other characteristics. Sperm concentration refers to the number of sperm per unit volume, whereas total sperm number refers to the total number of sperm present in the entire ejaculate. Motility discristings. Motility describes sperm movement, while morphology evaluates structural characteristics. Let's connect this with hormonal regulation. Sperm production is controlled by the hypothalamic-pituitary-gonadal axis. The hypothalamus releases GnRH, which stimulates the anterior pituitary to release LH and FSH. LH acts on Leydig cells to stimulate testosterone production, while FSH acts primarily on Sertoli cells and supports the process of spermatogenesis. Sertoli cells also provide structural and nutritional support to developing germ cells and participate in hormonal feedback mechanisms, including the production of inhibin B. Another high-yield concept is that sperm production and semen production are not the same process. They occur in different locations. Sperm are produced in the testes, while much of the fluid component of semen is produced by accessory glands. Therefore, disorders affecting sperm production and disorders affecting seminal fluid can have different causes and clinical consequences.
So remember this exam-friendly line. Sperm is a haploid male gamete. Semen is a complex biological fluid containing sperm and secretions from the male reproductive tract and accessory glands.
Sperm is the cell. Semen is the fluid that carries and supports the cell. And that single distinction forms the foundation for understanding male reproductive physiology, spermatogenesis, ejaculation, semen analysis, and male fertility. This is Zenvy Health, bringing medical science and human biology together in a clear, evidence-based way.
Analyzing these diagrams shows a significant breakthrough in cellular regeneration. We are very optimistic about the next phase of trials,